Andre Mardal. There is still an echo here, I think. Is it fine now? Yeah, okay, so now it's fine. So I work at Simula Research Laboratory. I guess you can see my screen even though I can't hear you. So we have been looking into the assumption of laminar flow in other types of flow in aneurysms and so on. And based on that work, we become curious whether it also applies to Chiari. So what I'm going to present here today is a recent paper published in Journal Biomechanics. It's called Numerical Simulation of the Pulsatile Flow in Cerebral Fluid Flow in the Cervical Spinal Canal of a Chiari patient, and it is by Anders Helgeland, me, Victor Houghton, and Anders Reif. I'm also going to talk a little bit about the work that a student of mine has been performed lately. This is not available just yet, but she has just submitted her thesis, so it will be ready in a couple of weeks or something for you to see as well. So I will just start. Okay, there is, yeah, okay. So our interest in Chiari started a couple of years ago, or actually some years ago. It was, I think, in 2007 or 2008, when Vic Houghton came to us and explained what Chiari was all about, and the fact that you developed. This is on the left-hand side here. You see a healthy person, and here you have the Chiari patient. And what we got interested in was, of course, the fact that a lot of Chiari patients also develop cysts further down there, and that seemed to us as a sort of a mechanical problem. So that is sort of why we have started to look into this, together with Vic Houghton, that you probably know. So the flow characteristics here is, of course, as you probably know, it's Newtonian flow. It's basically water. You have strong fluid structure interaction. Both you have deformation of the central nervous system, spinal cord, and so on, but you also have movement. You have a very complex geometry. You have pulsatile flow with a mean that is approximately 0. About 1 cubic centimeter goes up and down per every second, and the Reynolds number is typically smaller than 500, even smaller than that. So for that reason, nobody really has gone into the issue of whether there is transition because of this low Reynolds number, and nobody has detected it by chance either, I think. So of course, here you see an image of a healthy person and also a Chiari patient. And since we know that it's about the same amount of fluid that goes up and down, here you see this healthy person has a lot of space, whereas in the other one, the Chiari patient, there is a very tight area, in particular near foramen magnum here. So the velocity has to be larger, even though perhaps the flow is slightly less here. There's bound to be increased velocity. And also Victor Houghton and several others have done a lot of measurements demonstrating that the velocity in this area, or even further down, is a great deal bigger in Chiari patients than other ones. So of course, what we know, at least those of us with a background in fluid mechanics, knows that transition occurs when the Reynolds number is around 2000. That is what Reynolds discovered in his famous experiments a couple of 100 years ago. Of course, what he did was that he considered a straight tube, and he also considered pulsatile flow. And we are in a vastly more complex situation here. This is a Chiari patient, and you see that the geometry is definitely very, very complex. And of course, we also have pulsatile flow. So we cannot really use his reasoning in this situation here because it's totally different. So if we go back to our initial characteristics here, well, the Reynolds number is typically below 500, but actually transition may occur for Reynolds number 200 or something like that for application where you have flow around cylinders. And we have even seen that it occurs in about 350 in aneurysms. A lot of flow studies have been performed lately using CFD, and none of them have detected transition sort of by chance. But I don't think that is a big surprise really, because most of the studies have, or all of the studies have, reasoned that because of this low Reynolds number, the flow will be laminar, and I typically use dissipative schemes and also low resolution. Also, if you consider this in vivo with MR or something like that, the resolution might be too low so that you will not necessarily see it. Of course, you may also argue that, okay, this is a fluid structure interaction problem, so this might stabilize the flow. Well, this is a very difficult and delicate issue. You cannot really say that fluid structure interaction will dampen the threshold of transition. It may both stabilize and destabilize the flow. And the same applies to both the geometry and the pulsatility. So all of them may trigger or they may stabilize the flow. So based on this reasoning, we have done a rather extensive study of this in Chiari patients. So what you see here is a model of the geometry. So this is the model that we are interested in. Then we added some extension on the top and also on the bottom, just to allow for a simplified version of the boundary conditions. So in this application, we actually have 47 million cells. We have 1,000 time steps per second. I don't want to go into L plus and T plus. That's something for fluid mechanics people. They are very interested in numbers like that. But I just want to mention that one cycle here required 22 hours on, 22 hours on 48 processors. So in total, this computation here, it took about 1 month of time on CPU time. So it's a rather extensive simulation. I just want to show you a movie. This is a movie of the flow. Here you have a coloring of the velocity here, and you see in this movie it seems that the flow is fairly laminar. You see all the features here are relatively smooth. So what we do then is that we were not altogether happy with this result because it was so smooth. So we went in and looked in a little bit more detail, and then we looked at this area down here. And let me see. And this is what you will see in this movie here. Oh, okay. So this is a movie where we have colored the velocity going towards you, so it's in the x direction, so to speak. And what you see here is the velocity in the x direction. And what is interesting for us here is that you see these waves are forming. This is typical when you are at the transition point. So in some sense, this is not turbulent or transitional flow, but it's just below that it appears. We can also see that in graphs like here, you see that there are small wiggles. While the flow in the z direction, I mean the cranial cervical direction is the dominating one, and you don't see any wiggles there. But there are small waves forming in the XY direction, or the transverse direction. So this is an indication that in this patient there was no transition, or there was no turbulence, but it was close. So the conclusion in that study is basically that it's close, but this patient was a relatively nice geometry. So we have gone further now, and yeah, okay. Yeah, I just skipped this part. We have gone further, and in this case we have compared a volunteer, as you see here, and also a Chiari patient where you have very severe malformation. You see that there's a very tight region here at the foramen magnum. In this study, we also included the cranial part, so we included at least some of the ventricular system and also the subarachnoid space at the front here, just to and it's about 8 centimeters per second or something like that. In this severe case of Chiari, you see
Can
that.
I interrupt you? Kent, just a moment.
Yeah.
Can you go back about 20 seconds, because we missed what you said there. For some reason, your audio dropped out for about 20 seconds. Can you just go back one slide?
To this?
Yes, that's perfect.
Yeah, so I just start here again. So this is a study. This is not in the paper that I was discussing earlier. This is from the PhD thesis of Karn, so this hasn't been published yet. But here we compare one volunteer and also actually two Chiari patients, but I will only show one of them here. And the thing that we do here, we wanted to include some of the cranial parts as well. So we include some of the ventricular system, the aqueduct and so on, and also the subarachnoid space at the front here. And this is the Chiari patient on the right here, and you see that this is a very severe Chiari malformation. The area here around foramen magnum is very tight, and there's bound to be very little flow there, or very high flow resistance at least, and large velocities. So what we had here was that we had measurements somewhere below the foramen magnum at C2 or something like that, and we also have velocity measurements in the aqueduct, just like to mention that actually this flow in the aqueduct is slightly out of phase with the flow in the cervical part here. And here you see a representation of the geometry that we have computed on. So you see the aqueduct here, the cervical part here, and then you have the frontal subarachnoid space here. In the volunteers, we saw that, well, the velocities were very nice. The highest velocities were up in the aqueduct here, and here you had velocities of about 8 centimeters a second or something like that. But here around the foramen magnum and so places, and also further down in the cervical part, the flow is very nice. The other case here is the Chiari patient, and there you see that, well, certainly you have high velocities up in the aqueduct, but definitely you have the largest velocities down here associated with the narrow part around foramen magnum. And here we see that we have velocities of about 40 centimeters per second in our simulations. And you see that the flow down here is quite complex. So in this the simulations here, we weren't able to really resolve this properly like a DNS study, but definitely you see that the flow is highly complex. And we were kind of happy because this is a publication by Alexander Bunk and several other people demonstrating this vortices just beneath the foramen magnum, and it's similar to the ones we have here. So then I'm about to conclude. I would just conclude by saying that I think in disorders like Chiari malformation, the CSF flow can be quite complex. It can, I believe that it can even be transitional or turbulent. Fluid structure interaction may stabilize or destabilize the flow. For that, I don't have any idea. It's an open question that we need to look more into. Nerve roots and denticulate ligaments and so on may also promote transition, of course. Of course, common schemes, the schemes that most people use, they introduce stabilization and so on, or use simplified schemes so that transition may actually be delayed by the numerics. And that, I think, is, since we are in this area, we should be really careful about the numerics, because we might get totally wrong results if we are not careful with that. Okay, I think that was basically what I had planned to say here today. So thank you for your attention. And I hear the echo is coming back now, so I guess you can ask questions.
Yes, Kent Andre, thanks so much for your talk here. So I'm not going to ask questions first. Obviously, we have some terrible echo problems. What I want to do is go down the list, starting with Georgie, and I will just unmute Georgie and see if he has any questions. And also, you noticed I didn't have anyone introduce themselves at the beginning of the talk. So if you ask a question, try and say who you are. I'm apologizing for that. I missed the introductions, but I thought the echoing was so bad that it would be a mess to introduce everyone all at once. So Georgie, I'm going to unmute you. Georgie, do you have any specific questions?
Hello, Ken. Thank you very much. My name is Georgie Kuntges. I'm funded by Concord Chiari, and I'm interested in the development of And I have a small question for you. Great velocities. Can you tell us what this is adjacent to in terms of the involved?
Sorry, I didn't get that question. I only heard parts of it, so I think you have to repeat.
The region.
Sorry.
Arachnoidal spaces. Hello?
I'm sorry, I don't.
Kent,
Can you try again?
Kent, can you hear me?
I can hear you.
I think Georgie was asking, what neural structures, what actual anatomy are near these places where you're seeing these this kind of transitional flow.
So in the in the first study, we of course we didn't have any. There was no syrinx there at all, and we didn't include any nerve roots or things like that. So I think the transition occurred maybe around C3, C4, or not the transition, but the instabilities. There were also some suggestions for instabilities just below the foramen magnum. But also in that case, if we go back, sorry. So in this case, it was up here, sort of there was a tendency towards instabilities, and also down here. So I'm also a little bit uncertain about the actual geometry in this case. We did as good as we could, but the MR measurements were poor down here. Was that an answer to your question?
Not really, no. The region where the measurements were made, not where your model.
Yeah, okay. The measurements here. So these measurements were in the aqueduct and also at C2 or C3 here, whereas the instabilities were more around foramen magnum. And of course, in this area here, it's probably very difficult to measure the flow accurately. These
yes,
are very
yeah. Also for the analysis of all of that, structures are next to those areas, these two canals there that you pointed out.
Sorry, I didn't get that either.
So, Georgie, I think there's something very, it's very hard to hear you. Your voice is breaking in and out. So I apologize that I think we're just going to move on with some questions from some other people, and hopefully the internet will work better for their questions. So I'm just going to move on for now. One idea, Georgie, is you could call in by the phone number in the email, the message for the meeting. There's also a phone connection that might work better. I don't know. You could try that out. But for now, I'm just going to move on. I'm going to go with Mark. Actually, he's muted right now. So I'm just going to move on to Nick Schaefer. Nick, can you hear me?
Hello? Yes, I can.
So, Nick, why don't you guys, I know you have a few people there. Does anyone have questions?
Oh, yeah, Dr. Wall has one.
Hey, Kent, very interesting presentation. Nice to see fluid mechanics on Chiari. I wonder if did you check and see what the pressure drop that was created by that flow through that canal? Because then you can determine if you keep the millimeters of mercury pressure drop, you can see if it's physiologically possible.
Yeah, sorry. The pressure drop in this Chiari patient, both of them, or the volunteer is a reasonable pressure drop in terms of what you would expect. For this Chiari patient here, it was about 4 times as big as the volunteer. I can. I don't have the exact pressure drop just now.
Yeah, Ken, so when I saw the value of, of 40 centimeters per second, that's very high compared to what we see in vivo. I've seen it up to like 20, but 40 is very high. So we're curious.
Totally
that seems pretty high.
I totally agree. What we put in here was we had measurements in the aqueduct and also in the velocity down here at C2 or C3. So we had a pretty good idea of what the flow should be. So the huge velocities here is really determined by the geometry here that is very, very narrow. And of course it is a definite challenge to segment this area. We did as good as we could, but sure, I agree that these are very, very large velocities. But it's basically dictated by the geometry that we did as good as we could, because we know the amount of fluid that goes through this domain. So the inaccuracies have to be in the geometry. But we were careful.
So Kent, actually, if you look through, do you see the chat window? There's a chat window in the GoToMeeting panel. Like if you click on your GoToMeeting, and people are writing questions there. Maybe you could just go from the top. Georgie has a question. Do you see that? It's if you look in your little there's a GoToMeeting panel and there's a thing that says chat and a little button there that you should be able to push. And well, I can just read it to you, Ken. So Georgie had a question. He said, it will be very important to know the exact anatomical structures that are adjacent to the regions of maximum subarachnoid space flow, both for the patient and also from a developmental point of view. So he's just noting that specifying where those flow, the maximum flow or these flow alterations are happening relative to the anatomy would be good. So when we're writing our papers, we should document some of those things, kind of registering back to the original MRI anatomy image. As from what I understand, some of the in development of the brain, the way that the CSF moves and flows, this is thought to be guiding some of the neural, I don't know, the neuron growth. I've seen some talks like that. I think that's what Georgie's getting at. And probably he would type another question if he has more. I'm so sorry that the audio is very poor on this. So Mark Gooden, who's also online, he's also an expert in CFD. He has a question for you, Kent. He says, how much variation did you see over 29 cycles? Did the pressure and wave patterns repeat over the cycle?
Essentially, yes. There were very, very small fluctuations. So if you just look at it, it's basically the same.
Okay. Sarush, maybe I'm going to unmute you guys again. Or Nick, why not? I don't know if you want to use the audio or just type the question.
Yeah, we can try that. Can you hear me?
I can hear you fine.
Hi, I'm Surush Pahlavian from CCRC Lab. Thank you for the very interesting presentation. So I had a question regarding your yeah, Bryn, can you mute yourself? Sorry, I guess that would really help with the echoes. Thank you. Okay. Hello?
Hello?
Oh, okay, so you can hear me, that's good. So I had a question regarding your results where you were showing the sort of the transition in x direction velocity. So why do you think that the transition or the weird structures in the curves was happening in the x direction? Because as you said, the x direction is the transverse direction. And naturally, we expect the velocity to be smaller in transverse direction compared to the streamwise direction. So isn't it kind of weird to see that the transition is happening in that direction where the velocity is actually smaller? And also, how do you distinguish between the numerical error and the transition? I mean, in the curves, in the plots that you showed us, it was very similar to the pattern that we see when we have sort of the numerical destabilization of the results. So was there any way to distinguish between these two in your results? Or?
Yeah, so I'll start by answering the first question. So when you have transitional effects, it typically starts out as the waves in this x direction, because what happens then is that you get a mixing from the fluid that goes in a high speed with the fluid that is, for instance, close to the wall. So whenever you have some waves sort of in this x direction, you will get a mixing of these high velocities in the middle of the domain and the smaller velocities in other places. So when you investigate transition, it is this is the typical way of looking at it, because you see it more clearly. Transition will typically happen when you get a wave-like structure like that, mixing the different sort of laminar layers of flow, so to speak. As for your second question, it is hard to determine exactly the accuracy of the schemes and so on. What we have done here is to use a scheme that is has the same stability properties as Navier-Stokes, so to speak. So it doesn't add any extra stabilization terms to it. So in that sense, it is a scheme that is designed for picking up transitional effects. We also were careful about the numerics. I mean, we did standard grid convergence analysis and so on. But I totally agree with you that verifying numerics in this sense is a bit difficult, because you need extremely resolved simulations to do that. Now we were at 40 million cells, but of course we did refine the mesh in areas where we saw that there were big gradients. That was, I mean, this is not the initial mesh. This was an iterative method where we refined the mesh in areas where the flow was had great gradients.
Okay, so yes, partially. So you said that you use a very fine mesh, and you also refine it even more at the regions that you were expecting to see this high gradient. So what was the what?
Not where we expect them to be, but where the computations showed where they were.
Okay, okay. So I had another question. How did you check that, for example, because you were using a very fine mesh, so your, let's say, your cell, your representative length of one of your mesh cells should be very small, right? So wasn't it in the order of magnitude of your residuals or something like that? Because that might also introduce some errors into your calculation. So did you check for that also?
Yeah, we checked for I mean, the solution algorithms and so on, they were converged. So
I mean, for checking the convergence, you should probably use a residual criterion, right?
yeah.
So let's say that your residual criterion was 10 to the minus 6. And now if your length scale on your mesh is also on the same order of magnitude, that could have caused some problem. So my question is that, did you check for that?
Yes, we checked for that.
Okay. All right. I guess. Yeah. Another very small question. What scheme did you use to couple velocity and pressure in your Navier-Stokes solver?
Yeah, so this is based on do you see my screen here?
Yes, yes.
So I can let me see.
That's what I missed.
You had to do it years and years ago. I think.
Sorry, we haven't really written up the referred to the. I cannot find that publication, but this is not a typical Crank-Nichols nor implicit Euler scheme. It's a paper by Simon, and I mean this is a standard scheme for investigating transitional flow that we have been using. So it doesn't introduce any extra dissipation. Typical schemes that you would use, like semi-implicit schemes or something like that, would introduce diffusion. But other than that, I mean, the handling of the convective part, it is a standard incremental pressure correction scheme. But I can send you the paper on this specific scheme that we have been using. I can send you that. But it's not a standard scheme. It is a scheme that is designed for this kind of purpose.
Okay, that would be great. Thank you. Okay.
I wanted to come back to a question from Georgie. And he is saying, how do you reconcile the differences between your model and the measurement you made of the same Chiari patient? How do you compare them? So he's saying, how are these validated?
Yeah, so in our for our results, we aren't really able to verify them because we have a couple of transverse measurements. So we know sort of the volume of the flow, but we don't know exactly where these axial sections are. So we know that the flow is the volume of the flow is approximately correct, or there's some noise of course in the measurements, but since we don't have the exact place where these measurements were performed, we are not able to verify it in a detailed axial section manner. So we only know the flow, but the volume of the flow, but that should be correct in our. We don't have any better measurements than that, so that is we.
Kent Andre, are you online still?
Yeah.
Yeah, for some reason your audio cut out again. Everyone, I apologize greatly for the connection here. I'm not sure why it's so poor today. So Mark Gooden had a question that he typed in the window here, Kent, and he was saying to follow on to Sarush's question. He wanted to ask if you ran your simulation with single or double precision. And he said double precision could help with having very small cell sizes and trying to achieve very low residual levels.
Yeah, so this is double precision.
Okay, so it is. So I'm going to move down the list here. I think Sarah still has not spoken because her name starts with an S, and other people have names earlier in the alphabet. So let's see if she has anything to say. Sarah, are you there?
Hi, can you hear me?
Yes.
Yeah.
Great. Thank you very much for a very interesting presentation. I don't think I have any particular questions at this point.
Okay.
Kent, maybe Sarah, actually, would you introduce yourself a little bit so you know Kent can know who you are.
Sorry, of course. So I worked previously at Umeå University with the CSF dynamics and ICP analysis, mostly for hydrocephalus. Right now, I am at the Cleveland Clinic as a postdoc, mostly working in ICP. But I'm also very interested to great with MRI measurements.
Okay. I'm sorry, but I didn't hear all you were saying, but yeah.
Sorry, I don't know if I should try again or if we're just accepting that the audio is not gonna quite work today.
Let's accept that it's not working so well. I'm sorry again about that. So for me, I think from a big picture perspective, Kent, you presented a really detailed simulation, kind of pushing the boundaries of CFD applied to CSF. And obviously, our lab is working in this direction as well. We've been trying to do some simulations with brain motion, also including nerve roots. So we're beginning to get quite detailed. And Vartan's group has done work with the microanatomy with the arachnoid trabecula. And I'm wondering, Georgie had some really important questions. He's coming from the biological side, and he's wanting to push us in the direction of, seeing how these things translate to something useful. He's saying it in a nice way, I feel like, right? He's saying, wait, what are the neural structures near where these things are happening? He's trying to think of, the symptoms that we see. And so I think the validation of the work, which is one thing he asked about, and also Sarah was saying she's interested in, looking at CSF measurements to help us validate these kind of models. And then also beyond that is finding any utility to it. What can we do to improve our translation of this work in this simulation? It's so detailed now. We need to be careful to make sure that we're not going sort of too far before we do more validation and just translation of what's useful. And I'm wondering, from your thought, Kent, do you have any ideas of where we should spend more work? Is it more on validation of it? Should we be working on finding the clinical utility of it more, or where do you think the future should go in this?
From my point of view, our problem is mostly that we don't have good enough data. I'm sure you have access to a lot better data than we have, but that is currently our biggest problem. We are not able to validate these models properly. The second question, or the second comment, is of course that we should find this clinical utility for this. Definitely. For that as well, there is this question of data. I mean, we have not any good data showing what the consequences or the geometry pre-op and post-op. We don't have access to that kind of data. We only have this snapshot in time where we have a patient and we try to simulate that. So from my point of view, a stronger collaboration with both clinicians and also people doing and investigating measurements, MR measurements, and so is certainly desirable.
Yeah, agreed. So, it's kind of a vague question, I guess. It's not too specific what I'm saying, but we agree with this idea. I think Nick Schaefer. Does anyone else there have questions, or should we just should we end the meeting now? Go ahead, Nick. Anybody there have questions? All right, Kent, thank you so much for presenting today, taking the time to be with us. I apologize about the internet again. It's not been this poor on any meeting yet. So
it's likely my fault, though.
no, I don't think so. I'm not sure what's going on. But yes, I'm going to just end the meeting now, and we'll see everyone next month. Thanks. Bye.
Thanks. Bye-bye.